Welcome back. So in this lecture, we're going to talk about the molecular kinetics of aerobic cellular respiration. All right? Okay, let's get started. Firstly, let's talk about what's a simple idea, but the thing is it's reflexive in a way, so it can be a little bit tricky. So this is the idea of the redox reaction. All right? I'm going to use some pictures and a mnemonic device to help you clarify it. So one of the important, actually one of the most fundamental ideas involving energy transfer is the movement of electrons, right? So here's the movement. Electron, right? So it was here and it was there, and now it's in its new spot, right? So there's that. Let's talk about this idea. Have you ever heard of electricity, right? So electricity is the movement of electrons, okay? And we all know about electricity. It does some good stuff. It charges our cell phones. It keeps the light on, right? It keeps the ice cold. And so all of that still actually happens inside of our bodies. So the thing is there's two directions really that we're looking at for motion. So we're looking at oxidation, which means some different elements or molecules will lose an electron, right? So they'll be oxidized, okay? And it's called oxidation because normally what happens is there we go, oxygen. Okay, so this is easily observed in oxygen. So oxygen is an electron hog, right? So oftentimes it steals electrons, okay, resulting in this oxidized donor, okay? Here you go, here. It means that that poor molecule or element has been stripped and now ionized, right? Is in an ionized form now and is just minus their electron. So they've been oxidized. Oxidation is the loss of electrons. Reduction is the gain of electrons. It means a charge, see this charge here, has been reduced, right? So there's no charge here. So we went from a +1 charge to a no charge once we've reduced this molecule or this element, right? So the one that steals it is reduced. The one that gives it is oxidized. The gain of electrons is known as reduction. We call them redox because reduction and oxidation. One of the ways I hope you can help remember this is using a little mnemonic device. It's a little silly and the spelling's of course wrong, but I think it was helpful. So think of it as Leo, the lion, says grr, spelled wrong, grr is spelled wrong, but it's like this. So if you can remember Leo, you can remember loss of electrons, right, is oxidation, right? So then that means that the gain of electrons is reduction, right? So G-E-R, L-E-O, Leo. So I hope that's helpful. It's a little tough to remember the directionality of the movement of the electrons, but great. Okay. All right, so you remember this, right? This was when we were talking about how to get energy from some of our food. And the food that we're looking at in this particular semester is sugar, glucose particularly, okay? So we're going to start getting into these three arrows, these three different types of reactions. They're actually big reactions. They're entire metabolic pathways, right? So we're going to need some oxygen because this is aerobic, right? So oxygen requiring. And then we're going to use that to decompose, create this exergonic reaction. It's catabolic, right? Because we're taking something large and we're breaking it into smaller pieces plus energy, right? Energy, that's supposed to be an energy cloud, I guess. Anyway, of which is carbon dioxide, which we exhale, right? It's a waste product, as well as water and what? ATP and heat, right? Body heat. You were wondering this whole time what they were called, those three little arrows. Well, here's their names. This is what we're going to look at. There are 3 steps, and they happen in order, which is also important. Let's get a marker up. Okay, so the first step we're going to look at is known as glycolysis and pyruvate processing. The second one we'll look at is known as the Krebs cycle, or the citric acid cycle, right? Or the tricarboxylic acid cycle. That's all of it. Okay, and the third one, the big money maker, is the electron transport chain. Really good stuff there. All right, let's get into it. So when we're looking at this particular set of reactions, what's kind of interesting is how not efficient it is. It turns out evolution only really graduates C students. Now listen to me about what that means. It means that you don't want to spend too much effort to get the same job done. Then it's wasteful, right? That doesn't mean I want you to get a C in my class. Please get an A. We're not evolution, we're humans. We like values of 99.9%, right? So let's do well. But it does mean that this is not as efficient process as it could possibly be. It gives us just enough of what we need, really. So the total yield that we're going to get is around 32 to 38 ATPs per molecule of glucose. And there's some actual wiggle room when you think about it in a living cell, but in terms of the biochemistry, the experiments that we can do inside of the lab, it usually centers around 32 to 38 ATP. It's not a lot for every molecule of glucose. So it's also only 34% of all of the potential energy that's in there. So we're only getting like a little more than a third. That's okay, because apparently we don't need more than that. And when you see the process, I think you'll understand why a little more. The other thing is just remember the things that are produced. Water and CO2 along with that ATP, right? So those are the other byproducts. So we talked earlier about cellular respiration and how you need one really important organelle, right, at least in eukaryotes, to do it. So the important structure is ultimately the membrane, but an organelle with a lot of membrane is going to be the mitochondria. So some of these items are going to happen on the inside of the mitochondria, and one important step is really going to happen outside of the mitochondria, where the mitochondria live. They live in the cytoplasm of the cell, right? So you remember how a cell looks. So here's the cell, and there's a nucleus, and inside the cell is all that cytoplasm, right? It's the jelly-like substance of the inside of a cell. And so in that cytoplasm is where all these little mitochondria are swimming around, okay? So again, let's look at which processes are where. One process, the first process, is glycolysis pyruvate processing. That happens in the cytoplasm. The next one is citric acid cycle, number 2, also known as Krebs cycle or carboxylic acid cycle. Wow, tongue twisters. Okay, and then 3, the last one, is going to be the electron transport chain. Okay, also oxidative phosphorylation. And we call the final step of it, where we actually make the ATP with this really cool enzyme, chemiosmosis. We'll get into those names a little bit later. Don't stress yet. I'll explain them out when we get there. But in the meantime, I want you to think about something, like a big picture idea. So pretty much eukaryotes and prokaryotes were the same for a really, really long time. And then an evolutionary step happened. One of those evolutionary steps was likely the acquisition of these mitochondria. But one thing that prokaryotes and eukaryotes have in common is the ability to split sugar. For the most part, all of us can do this. That's why that part happens in the cytoplasm. That's actually the only part that we can do on our own, right? All the rest of the steps, like getting those higher numbers of ATP, have to happen on the inside of the mitochondria. They hold the secret. They're specially evolved to do all that. So if you took all of the mitochondria out of your cells, you'd only be able to rely on this first step of glycolysis to get any ATP, of which there's not a lot. Look, it's only 2, right? So that's something really important to keep your head around. The other thing that's really important to keep your head around is look how much you get from the mitochondria. What do you mean? We stepped it up, right? Quite a bit. 30 more than we would normally be getting. Can you see why we kept these mitochondria around now? Really lucrative. Good idea. Okay, so yeah, let's get going on the next steps. So I want you to be aware of, you can't just do this on your own. Some of you have noticed that you feel a little bit better after you take your vitamins, for example, right? Have you ever wondered why? What nutrition really means? You can't just live on sugar, right? Actually, you need a few other things from the environment. Important things, usually like charged types of ions or missing components that fit into an enzyme. So those things that we're going to look at are cofactors and coenzymes. Cofactors are usually small. Oftentimes they're ionized, right? So they have a charge on them. They're things like copper, zinc, iron. So those are some metals that can be slightly charged. Basically what happens is you plug those into the active site. If you don't remember what an active site is, you can go back and watch the lecture on enzymes, right, and enzymatics. And they'll plug into the active site or the working site of an enzyme, and they act kind of like a little magnet in a way, and they attract that specific substrate. Without this particular cofactor, we're not as specific as we can be. Also, we're not as aggressive for grabbing the substrates we need in order to break them down. Let's move on. The next one is bigger, the coenzyme. Whoa, that already sounds bigger, right? So this is going to be in the category that we think of like vitamins being in. Okay, they're bigger, they're organic, which remember what organic actually means. It's a carbon to hydrogen bond. That's what organic is, okay? It's not just pesticide free. In a chemical sense, it's carbon hydrogen bond. And when we're talking about cellular respiration, we're going to talk about two really, really important types of coenzymes, right? So they're going to be nicotinamide. Oops, hold on. Let me get my highlighter. There you go. Nicotinamide adenine dinucleotide. I said that 3 times fast, right? Just think of it as NADH, and when it's ionized, NAD+, okay? The next one is going to be flavin adenine dinucleotide. Flavins, right? So this one is going to be FADH2, 2 hydrogens, and when it's ionized, it only has the 1. It's not really ionized as much, but we'll see what that means in a bit. And the last one we deal with is coenzyme A. All right, we're going to use that to modify an early stage component of these metabolic cycles. So these coenzymes are going to help complete an important process in the cell without being enzymes themselves, right? So these are obviously not major proteins, but we're going to use these smaller molecules to complete important functions. And in this case, these two are going to transport electrons, right? That's their main one. They're also going to move around hydrogens, right? Which help us manipulate electrons, right? As much as the electrons help us manipulate where the hydrogens go, right? So we remember how magnets work. So we have electrons, negatively charged, and protons. Okay, when we think about these hydrogens, I want you to think of them as they're ionized hydrogens, right? So it's like proton and no electron, right? That's why it's got a plus charge, so it has no electron. So that's why it's this polar opposite here, right? So North Pole, South Pole, kind of like a magnet. So they're going to manipulate each other in space in chemical reactions. That's important. So I just want to underscore this fact really quickly. I know that I had it on a smaller slide, but just in front of us, and we talked about it a bit, but just please really remember these. These are important coenzymes, NADH, FADH, and the coenzyme A. Please remember that. Write them down. Important reasons you need to know them: they transport electrons, and they also transport protons. Both are types of ionized particles, subatomic particles, right? So, how can I think about this? I guess one way to think about these would be almost like little delivery trucks, right? So think of them like a little delivery truck. Okay? And sometimes they're going to have electrons and hydrogens to transport. And sometimes after they drop them off, these are some sad trucks. I'm super sorry, but let me do my best. And sometimes after they've dropped them off, they're going to need to go pick up some more because they'll be empty. They're going to run these back and forth through different parts of the cell, and that's going to power some pretty important things, especially this one big enzyme called ATP synthetase. ATP synthetase makes the majority of the ATP that we make in the mitochondria. All right, so that's them. They're little shuttles in a way. So we looked at the idea of redox reactions, right? And how they are important for understanding how electrons move. Oxidation, reduction, who has an electron, who just lost electrons, right? So that's important. We also looked at some things like cofactors and coenzymes, right? So those are also important. We learned about 3 really important ones, 2 which are going to function in major transport, right? Just like these little trucks here. So we want you to keep those in mind. They're important tools or resources in our production of ATP. All right, we'll see you in the next lecture.